Texas Lutheran University.
We are excited to welcome an alum of TLU, Danny White, Dr. Danny White, to our school. To give you a little bit of background, he did a tour of Iraq as a Marine. Then he got his associate degree in science at Victoria College. Then he came to TLU, was actually on the environmental track, was kind of interested in dentistry, but then his plans changed because he went to get his PhD at the University of North Texas Health Science Center in Fort Worth in physiology. Then he did a postdoctoral fellowship at the University of Illinois Chicago in integrative physiology laboratory. And now he is at the University of Houston, Victoria, where he is the Director of Graduate Studies and the Pre-Health Professions Advisor. So let's welcome Dr. Danny White.
Okay, so over the next 3 hours, I'm going to be talking to you. Yeah, right? They told me I have 15 minutes to talk, so I'm going to take 45, okay? But what I'm going to talk to you about is something that I'm interested in and something that I do all the time, or at least used to until I moved down to Victoria. Now I'm trying to get my lab back going again, but that's autonomic function and research in autonomic function. I like to make waves in the world, so I said, no, that's not right to all the previous data, and I changed it. And that's something that I suggest everybody try to do, make waves in the world, right? I changed it. So as she said, I've got associates, BS, PhD, and I did a postdoc in Chicago. What I'm trained in is integrative physiology, and that combines cardiovascular physiology, autonomic neuroscience, and exercise physiology. I dabble in a little bit, not so much training you and making you better, but seeing what exercise does acutely to the cardiovascular system and to the autonomic nervous system, right? So that's me in a nutshell. But let's start. Let me talk about the autonomic nervous system. One thing I learned from Dr. Squires while I was here is that you always start off with a history lesson, right? So way back in about 170 AD, is that true? Always start with a history lesson? Yeah. And then he tells you about marching? Yeah. So way back in about 170 AD, Galen was doing anatomical studies, and he was looking at nerves that come out of the back of the brain. And those are called cranial nerves now, but he was looking at those and he figured, well, these nerves are hollow, and when the brain gets disrupted, those hollow nerves transmit the feelings from the brain down to the other organs. So the other organs are sympathetic to the brain. And this was, well, kind of the start of the sympathetic nervous system. Even though now we know that those nerves he was looking at are part of the parasympathetic nervous system, this was where that kind of started. Then, a couple years later, Eustachius conceptually separated the two sides of the autonomic nervous system, right? The vagus, or the parasympathetics in the vagus, and the sympathetic nervous system, right? Then Willie figured out that, well, those parasympathetic and sympathetic nerves, you don't control those. You don't control those the same way that you control maybe your fingers, your arms, or your legs. Those are automatic, and that's why it's called the autonomic nervous system. It's the nervous system, the side of it that you don't actually control. A couple more years later, they started classifying these neurons, and they figured out that these neurons of the sympathetic nervous system are unmyelinated, and they don't really conduct very fast, and things like that. So they were doing classifications of those, just refining what we know about the nervous system. To actually figure out how they work, Claude Bernard in 1851 figured out that if you cut a nerve, one of the sympathetic nerves, to a muscle bed or to a vascular bed, those vessels dilate. So there must be some kind of a signal going down that's releasing something at the end that causes constriction, right? About a year later, another physiologist figured out, hey, if we take that cut nerve, we put some chemicals on the end of it, we can cause those vessels to constrict again. So now we're starting to get into, okay, this is really how these nerves work, and this is how they affect the cardiovascular system. Well, let's get into it. The brain is a black box. or at least it always has been, and it was definitely a black box back then. It was just a bunch of tissue sitting up in your head, and nobody really knew what was going on inside of it. We knew these nerves came out of it, right? And so they started figuring out, okay, well, what do these different nerves do? What do they cause? What can we poke in the brain in order to find out, oh, this one makes your heart rate go up, this makes your heart rate go down, this makes your blood vessels constrict, things like that. And Volkmann figured out that changes in heart rate and blood pressure with normal respiration and muscle contraction, he defined that stuff. So whenever you breathe in and out, your heart rate goes up and down like this, right? Whenever you start to exercise, if you squeeze something really hard, your blood pressure starts going up, right? These are all neural reflexes. And it was a little bit later on that Johansson noticed that heart rate increase whenever you start exercising is almost immediate. But they still hadn't figured out, well, how does all that work from the brain? Sometime between here and here, Murray noticed kind of off over here that normal changes in blood pressure and heart rate are inverse, but related. So if your blood pressure goes up, your heart rate goes down. Blood pressure goes down, your heart rate goes up. And that's the arterial baroreflex, right? Baro meaning pressure, reflex meaning reflex. So whenever your blood pressure goes up, your heart rate goes down. So now we're starting to talk about, okay, autonomics, right? So now we've got the heart, blood vessels, baroreflex. That's going back to the brain and coming out. So guys wanted to see, okay, well how do these things interact and what else could possibly be going on, especially when you start exercising? That was Krogh and Linhardt and Danes. And what they said or thought through some really pretty neat experiments was, well, okay, at the same time that we decide to start moving our legs, that's when the heart rate starts going up, that's when the blood pressure goes up. There must be something in your brain that's telling your legs to move, but at the same time working through the autonomic nervous system in order to make heart rate go up, in order to make blood pressure go up. Right? Pretty cool, huh? Well, they coined the term central command. That's the central command that's commanding all of the things in your body to take place at the same time. So that's still going into the black box, which has now been labeled the medulla. It's not the whole brain anymore, it's just we're centralized to the back part of the brain, okay? A couple more fellows were trying to figure out, okay, well, what other reflexes and things are going into the brain in order to cause things to happen when they come out? And they discovered that if you work your muscles really hard, like say a leg, work it really, really hard, and then you dip it into a vat of mercury in order to cut off the blood flow back out of the leg. Yeah, I know. 1937, mercury was okay for you, right? It's changed since then. Dip it in. So what they've done is they've taken all of the metabolites and things that you build up while doing leg exercise, cut it off so that it stays down in the leg. We do this now with rubber bands, right? We don't dip you into mercury anymore. But they figured out that that's the muscle heart reflex is what they talked about, because your heart rate would stay up and your blood pressure would stay up until you pulled your leg back out of the mercury and let those metabolites flow. They also noticed that, well, if we just start bending these muscles back and forth, we get some of the same responses. And those are the mechano and the chemo reflexes that are part of what we call the exercise pressor reflex, right? Still a black box, though. Still a black box. Eventually, people doing very elegant animal work had figured out how to open up that black box and figure out where all these inputs connect inside of your brain in order to make these oldest of neural functions that we've known about for, ever, how it makes them work and how we get these reflex loops back through just different small groups of neurons inside of the brainstem that all connect and interact with each other. Lots of the work was done in animals. Most animals don't stand on their hind legs. So a lot of the old animal work didn't take into account venous blood and the fact that we stand up. So the cardiopulmonary baroreflexes were known but not thought of as very important until, oh no, we start measuring this stuff in humans where it's actually applicable. We have to take into account cardiopulmonary baroreflexes. And those are some pretty cool things that I like playing with. And that's where things like lower body negative pressure and things like that come into play with those cardiopulmonary baroreflexes. But overall, this is the picture that we think about whenever we think about neuroreflexes. And we think about things like the central command and the exercise pressor reflex as feed-forward accelerators, and both the baroreflexes as feedback inhibitors, right? Okay, so that's the broad picture, okay? Well, Me, I like to make waves. And while I'm sitting there doing my PhD and I'm looking at millions and millions and millions of data points trying to figure out how all these things come together and how the baroreflexes interact during exercise and things like that, I noticed one thing, and it was that what I learned in exercise physiology didn't seem to be true whenever I was trying to actually use it in the lab. I was like, well, that's kind of strange. What's going on here? Why is everything that I got taught a little bit different? It's not working like it's supposed to, okay? Anybody seen that? Who's an ex-phys person in here? Any ex-phys guys? Yeah. See, Dr. Squires is pointing y'all out, and I'm looking over and I'm like, yeah. So, what this says, and this is what exercise physiologists, kinesiology majors, people like that, this is what you learn about the autonomic nervous system and the interactions with heart rate at the onset of exercise, right? Up to about 100 beats per minute, you know, it's still a very low oxygen consumption. You get a withdrawal of the vagus. The vagus goes away, your heart rate goes up, right? That's one branch of the autonomics. And then from about 100 beats on, and whatever exercise it takes to get you there, the rest of that is due to the sympathetic nervous system, okay? So our total acceleration of heart rate all the way up to max goes, whoop, parasympathetic, and then whoop, sympathetic. One and then the other, and they've been teaching that for years and years and years. And I said, well, no, that ain't right. That ain't right. And me, I like making trouble, so I made sure I told them it ain't right. One thing that I know I can do, right, so that graph was made based off of pharmacological studies to where they would go in and they would block with atropine and they would block with propranolol, things like that, and figure out, okay, well, where's the heart rate here and there and everywhere? But whenever you do pharmacological blockades, you have to wait till the drug wears off. You can't do anything else. So once you start that drug, you can't collect data any further and call it not drug induced, right? Well, with something like this, I can take, and I know that by stimulating your arterial baroreflexes, I can change your heart rate immediately, beat to beat, and I know that it is always changing because of oscillations in the parasympathetic nervous system. So I hit you with a pressure, parasympathetics withdrawal, your heart rate goes up. I hit you with a suction, right? This is just using pressure and suction. Suction, parasympathetics get activated, heart rate goes down immediately within one beat, but then comes back to normal. So I can do this at every stage of exercise and tell you whether or not your vagus is still active. Cool, right? It should stop being active at about 100 beats per minute, right? That's what Ralph said. That's what your ex-phys book said. Well, it's not true. These are very basic and hard to look at from way back there, and they don't really show the actual resolution of how many, 8 beats per minute change in heart rate within one beat is huge, right? So what you can see here is that if this is workload going up and this is max, right, this would be the reflex, how much I can actually turn your parasympathetics back on through next suction, okay? Working all the way up, and it's actually still functional all the way up to the end. I can also do the opposite and all the way going up, turn them off, meaning that there is some parasympathetic activity that is still functional, right? It's just mostly quiet when you exercise, okay? And that's all parasympathetics. Well, you can't just look at reflexes and make conclusions and publish in a good journal. You have to look at steady state interactions and using some pharmacological blockades, which were glycopyrrolate or metoprolol, right? So a muscarinic blocker and a beta blocker. This is normal, all the way up to max. If I take away the parasympathetics ability to bring your heart rate down, you go up to that 100 beats a minute, which is where they got their number from on that original thing, but it still goes up, okay? Meaning that the sympathetics were actually causing that increase. But when I take the sympathetics off, heart rate still goes up, meaning that the parasympathetics were actually contributing to the rise in heart rate all the way through exercise, okay? The one thing that they did get right in your exercise physiology book is that heat, right? Heat does cause some of this up because if I block both sides of the nervous system, you still get a little increase, but it's more like this. Okay? Pretty cool stuff. So I made this very complicated graph, and I went to go turn it in, and my PhD advisor, Dr. Abrams, said, no. Nobody's going to look at that. Make it simple. So I made it simple. Right? But I made it so simple that people got confused. Okay? Because that is probably the most elegant concept you've ever seen, but you couldn't tell. Because it's not actually, hey, does this one contribute 20 beats per minute to the resting heart rate? It's not, does this one contribute 80 beats per minute to resting heart rate? No. It is taken from, if you are at a resting heart rate, the percentage that's actually contributed by each side of the nervous system. They don't contribute equally, but to that steady state, it actually is. And as you go up, even though the overall change is less on which way I can go, they still have contributions, right? So this is fun, exciting, cool stuff. So using the idea of reciprocal antagonism, which we know does exist within beds of neurons, especially those dually innervated by sympathetic and parasympathetic nervous system. When the sympathetics kick on, they inhibit the parasympathetics. And when the parasympathetics kick on, they inhibit the sympathetics, right? A lot of people have drawn these seesaw diagrams. I figured I'd draw one too. Sympathetics, right? At rest, sympathetics have some kind of a steady tone for the most part. We'll just call that sympathetic tone. And then we'll call this parasympathetic modulation. That's the ability of your parasympathetics to move your heart rate up and down at a given sympathetic tone. We start exercising a little bit, and guess what? Those cardiopulmonaries actually kick in, and they inhibit sympathetic tone. Your sympathetic nerve stops firing as much when you have just a little bit of exercise going, right? You're just moving your legs. So the parasympathetics really come out. And I can modulate your heart rate up and down on a beat-to-beat basis using neck pressure, neck suction, like really hardcore when you're just barely exercising. That's pretty neat. Nobody thought that happened, but they'd never measured it that close to rest before. Now, when I get you really going, okay, and I'm wearing you out, it says, greater than 120 beats per minute. This is more like 150 beats per minute. This is cycling for 20 minutes at 150 beats per minute. Well, I've activated those exercise pressor reflexes, and I've activated that central command almost maxed out, right? So I'm forcing those sympathetics. They're actually overpowering those cardiopulmonaries, and I'm forcing those sympathetics to come out. And we see that as a decrease in parasympathetic modulation. So reciprocal antagonism shows very well in these concepts, okay? So that was me changing the world. And what I ended up doing was I changed that original picture that you all have in your book to look more like that. All right? This is kind of like borderline plagiarism, especially since I published it, but it's kind of like just taking the picture idea and making it my own. And since it was peer-reviewed and things like that, I don't think I'm going to jail for it, But that ended up in the Journal of Physiology. And what it does is it splits those into steady state and reflex contributions, right? So the vagus, down here at lower exercise, we see that the sympathetics kind of get weaker. Vagus comes out, right? And then as the sympathetics get bigger, the vagus stops having its effect. You have to think about it, if you're thinking about reflexes in reflex fashion, you can't think about it as steady state. But at steady state, it looks more like this, to where both sides are always contributing, and they just kind of change their roles closer to the end and their influence. That was the first paper I ever published. Changed the world, right? I figure I got to keep the waves moving, right? And I was specialized in a technique called microneurography. And microneurography is where I take a microelectrode, okay, and I put it into a nerve bundle and measure electrical oscillations inside of that nerve bundle. So if you look up here, you can see this would be a leg, right? That would be my preamplifier. I put a reference electrode just under the skin inside the muscle. And then I go in with an active electrode that is insulated all the way down to a 5 micromillimeter tip, and I put it into a nerve bundle. That's my electrode, is ultrasound. My electrode, that's a nerve bundle. Now, nerve bundles have many different types of nerves inside of them. So imagine trying to put the head of a needle onto one strand inside of a piece of yarn. And that's what I'm doing right there. And whenever you get the correct nerve that you want, it has firing characteristics that look like this, right? Each one of those actually fires with a heartbeat. Sometimes they don't fire at all, but it is gated by the heartbeat, right? And it is gated by blood pressure pulses that go to both your aortic baroreceptors and your carotid baroreceptors, right? The reason I actually did this paper, because I had done this so many times, and I kept looking and trying to compare my data to everybody else, and I couldn't. I could not compare my data to everybody else because everybody who was trained this, we started measuring this back in the 1970s or something, and I say we, like that's whenever it started happening, was in the 1970s. And so you have like the grandfather coming down, and I'm pretty much the great-grandson of the guy who brought it over to America, okay? His name was Alan Mark. And I'm pretty much the great-grandson of him because we don't train very many people to do this. It's too hard of a technique. It's not worth the time to actually do it. But if you have one guy who can do it, it really improves the paper, right? But we were all measuring it different because everybody had come up with their own analysis techniques and things like that. So I wrote this paper to get us all back on the same page. A lot of people didn't like that. A lot of people said, no, you're going against what Gunnar Wallin said, and this was the guy in Sweden that first did it. He said, no, you're going against what he said. He said, you can't do that. And I said, yeah, but he didn't have 8 gigs of RAM and the ability to look at 8 million data points within about 5 minutes. He did it all on paper, and he took calipers, and he took rulers, and he took his pencil tracing and things like that, and did it, and then figured out, okay, well, now the signal's moving up and down, so we have to figure out a way to do this, I'm lucky. Boop, done. I can push a button, normalize signals, first flatten out signals, then normalize signals, and then measure different activities, right? So these numbers are tiny. Y'all can't see those, right? The way that I do it is here. The way that some other people do it was there. We'd still get the same numbers for burst frequency, which is how many times a nerve bursts per minute. Whenever we normalize it to the number of heartbeats, we'd get the same answer for the number of bursts per heartbeat, right? The problem is if you're only counting frequency or you're only counting number per heartbeat, you're not taking into account how big that burst is. And a tiny burst that I'm measuring probably doesn't put the same amount of norepinephrine out into the system as a big burst. At least that's what I think. Therefore, you have to take into account the size of the burst along with the frequency. So the real question was, how do we know how big each one of those bursts are? And how do I know that my burst that is this big is the same as your burst that is that big? And I actually don't have an answer to that. But I proposed that you find spontaneous bursting, and this comes from years of me sitting there looking at these little signals, and I noticed that whenever you have a premature ventricular contraction and you have this long diastolic period after a premature ventricular contraction, you get a nerve burst that goes up and plateaus and comes back down. And that nerve burst is always the same size when it goes up, plateaus, and comes back down. And I also get spontaneous bursts, like this one, that are the same size as that premature ventricular contraction. So I figured that is the functional top. That is as big as a burst can possibly go whenever I quantify it. So I proposed that we take and we set all this noise to 0, and then we find the biggest spontaneous burst and set it to 100. Makes sense. Lots of people do that. Lots of people do it now. Lots of people quote my paper based on that. But there was one guy that said, no, you can't do that. So why not? He said, no, you can't compare nerve traffic. You can't compare nerve traffic. And we still come up with the same general answers. And I said, well, yeah, we still come up with the same general answers, but I want to be able to compare my stuff to yours. Well, he does it. So imagine these are 2 sets of the same nerve data. Mine is normalized to 100 arbitrary unit max. He sets the 100 arbitrary units to the mean of the burst signal, and this still blows my mind that he can't mathematically comprehend this. The less bursting you have, the more time that signal spends at 0. Therefore, if you set the mean to 100, the lower the bursting, the more you think the mean happens. So he's actually normalizing total activity and saying that all of these signals equal the same amount, right? Where I'm saying, nope, the one that bursted more often and bigger, it is bigger, right? Anyway, that's a fight that we still have. He's up in Michigan, and I almost slammed him through a poster at a national conference. That's my rant, sorry. Anyway, what I did was I put out guidelines that now everybody is starting to use, right? I like changing the world. I like making waves, okay? Even he has said, yeah, I do it that way now too. But he doesn't. We're friends, I guess. Frenemies, that's still a word, or did I date myself? Frenemies. And I also talked about maybe filtering the signals in order for better quantification, easier quantification. So then I'm up at a postdoc in Chicago in the Integrative Physiology Laboratory, right? I have gone from doing all these invasive drug studies and poking nerves and all kinds of stuff to a place where they deal with diseased populations. And diseased populations that you can't actually put a needle into. So in Down syndrome, they are not going to let me take a microelectrode and put it into the peroneal nerve of a child with Down syndrome. You just can't do it. You cannot get consent, and it's not a necessary medical procedure. So there's no way I can do it. So we have to figure out ways to quantify the autonomic nervous system without actually poking somebody, right? Yeah. Well, they've been doing it for years. They just measure your heart rate and the fluctuations in your heart rate going up and down. And it's called heart rate variability, right? Heart rate variability is one of those push button things that you can do, and you get an answer, but most people don't actually know what the answer means. But through no fault of my own, except for I signed up to go there, I became an expert at heart rate variability. And they'd been using it for years and years and years, and guess what I did? I decided I was going to make waves again. And these are guys who have seriously been publishing for years in these diseased populations based on heart rate variability, and I said, you're doing it wrong. Except this time, they didn't not like it. They said, well, let's make it better. I had one of the best postdoc mentors ever, a guy named Bo Fernhall. He wrote the book on cardiovascular control during exercise, the other book on cardiovascular control during exercise. And he was always open to a new idea. And so he facilitated my endeavors in trying to figure out how heart rate variability works. The heart is not a metronome. Those would be milliseconds, right? And beat to beat, your heart rate changes, okay? Right? And so we can take these beat-to-beat oscillations, we can plot them over time and make a tachogram, right? And a tachogram looks kind of like that, right? And then we can take and use fast Fourier transform and break it into different frequencies. So within this signal here, we can pull out a very low frequency signal, a low frequency signal, and a high frequency signal that sits between parameters of our choice, right? Okay, so just to make that a little bit easier, if I have something at 1 hertz, it shows up at 1 hertz, 2 hertz, it shows up at 2 hertz, 3 hertz, it shows up at 3 hertz, and if I mix them all together through, hopefully everybody has learned about summatory and cancellation of waves in physics, right? It looks like that if you add all those together, right? Everybody's got that, right? And then what we do is we take these little spikes out of it and we make conclusions. Make conclusions. Well, I told them, no, your conclusions, they're not right. And I didn't say it like that. I didn't say, oh, your conclusions are not right because I needed to keep my job and things like that. But I said, hey, you ever thought about what happens when you try to compare people with 2 totally different heart rates? Because somebody with a higher heart rate actually starts off with a smaller gap between heartbeats. I got 2 groups of individuals, young, healthy college students, and I sat them on bicycles. And some of the college students, when they sit on bicycles, the more fit ones actually had lower heart rates, down in the 50s when they're just sitting on top of a bicycle. The more unfit, but still weight height matched, right? But we did exercise testing on them, so I know they were less fit, had heart rates right up in the high 70s and 80s, right? So now I've got these 2 groups that are just perfectly separated out as, whoa, these guys are below 60 beats per minute and these guys are above 75. Let's compare their heart rate variability through controlled breathing, right? Your heart rate goes up and down as you breathe in normally. We learned that in like slide number 2 or something like that. So, but as you go in and out, so if I pace your breathing, I should be able to pull some oscillations in your heart rate out based on where they were at your mean heart rate, right? This is about to get really intense and boring, okay? It's going to start being math talk, and I don't like it, even though it's in my head. So we had 4 conditions for this. We had rest. Okay, we have rest. Well, what about if we match these 2 sets of people with their heart rate? And I said we can put you on a cycle ergometer and get you to move your legs with barely any workload. And I know I can bring your heart rate up to 80 beats per minute, and I know I can take this other group up to 80 beats a minute, and I have you matched at heart rate. Okay? There's some problems with that have to do with some of these autonomic reflexes in exercise, but for the most part, we're not actually working them very hard, which I showed you with my first wave, right? I can also work you at a relative workload, each group, which would bring each one of them up from their resting heart rate 10 beats per minute. So we've staggered like this, right? Then that third group, just to make sure that we're covering all the bases, I just cycled them at 20 watts, which is nothing, at 60 RPMs, okay? 20 watts, 60 RPMs for somebody who is fit doesn't really change their heart rate. Someone who is unfit, it actually made it go so that was kind of a surprise, but you know, I can really look at you and kind of giggle in the back of my head based on how well you pedal a bicycle. That's what you get from working a postdoc at an ex-phys place. We started laughing at you for that. What we saw was that at rest, the low heart rate group, right, those guys down in the 50s, right, had a much higher, we'll just call this mean beat-to-beat change in heart rate, okay? That's the RMSSD, the root mean squared, the squared deviation, right? Basically, the mean beat-to-beat change in heart rate. It was much higher. That means their heart rate fluctuates much more every time it beats, okay, compared to those high heart rate people. But when I put them both at 80 beats per minute, they're exactly the same. Ooh, maybe we should start testing people with non-invasive autonomic function tests at a known heart rate. Yeah. Whenever I staggered them based on a relative workload, still totally different. And at 20 watts, totally different, right? Significantly different. Whenever we start looking at power spectral densities, it gets a little bit tougher, okay, and that's that frequency analysis. This is actually time domain analysis, this is frequency domain, and because the units are skewed and exponential, you have to log transform these data back down to normalize the data for statistical purposes. The trends are the same as they were up here, right? The trends are the same. Statistically, I couldn't find differences in them. And that's one of the problems with heart rate variability and why you really shouldn't use it as a clinical measure, because I can look at that and say, yeah, those 2 groups, they look different. But should I diagnose this person as something is wrong when they're not, maybe statistically, and maybe they're showing no signs? No, you shouldn't, right? So that's why this is a hard clinical measure. The high frequency is going to show our respiratory modulations, and the low frequency down here is going to show those barrel reflex modulations. So these happen a whole lot slower, and they're due to changes in your blood pressure that actually change your heart rate in the other direction, right? Yeah, keep that in mind for the next one, because now, okay, if we physically normalize heart rate, I find that they're the same. Not everybody has a heart rate that's 80 beats per minute. But what about if we use math? So within a person, so now we have our low group here and our high group here, and within a person, I mathematically normalized their 80 beat per minute to their resting, right? And I did that just with proportional scaling of that tachogram, right? And what I found was, and all of these little things say no difference. Yeah, this is where we rely on statistics, right? Because those do look kind of different, but statistically, no different, no different, no different, no different, no different. So that was in that time domain. That's in the high frequency domain, right? So that's the breathing. If they're no different, what that says is that we can mathematically normalize these things, right? We can mathematically normalize these things to talk about heart rate changes per respiration. But over here in that low frequency where we talk about baroreflexes, it didn't work. And it was because those are on a totally separate set of neurons. Those are the sympathetic neurons feeding back to the parasympathetics, where all this stuff is just parasympathetic. So our mathematical normalization didn't work on the sympathetic side of things. And that's pretty cool. The problem is, you write a paper like this, and a guy who's been using this his whole career, and you tell him, hey man, you maybe want to be matching your heart rates, they don't like it. And then the guys that don't care about this stuff at all are the other set of guys, and they're like, it's heart rate variability. They don't like it either. They don't care. So you can't get that published, and I've been trying to get that published, and I'll do a few more. I think once you put it in 10 times, you just kind of throw it away, right? You kind of throw it away. But so that was a project that I did with one of the PhD students up there, and it drove her crazy. She actually dropped out, and she's a bartender now in Virginia. So, yeah. You gotta be able to wrap your head around some stuff like that if you want to get the PhD, right? We also looked at blood pressure variability just to see if anything changed. And, you know, of course, with the exercise in all groups, systolic blood pressure significantly rose. Diastolic blood pressure didn't really change in either group. There were some interactions there. Nothing is actually really interesting there. So y'all, the neck collar thing, right? That made your heart rate change? You can do that without a neck collar too, using mathematics, right? And what we can do is we can beat to beat measure your blood pressure and beat to beat measure your heart rate. And we know that there's about a 1 second delay from that blood pressure change to that heart rate change. And we can say, well, if it changes 3 times in a row in this direction, 3 times in a row in that direction, we call it spontaneous baroreflex, and we can calculate that stuff, right? So it's there. It's fun. You can see that in the resting population, we're back to low heart rate people, high heart rate people. Low heart rate people had a higher spontaneous baroreflex, right, using cross-correlation analysis. This is me grabbing for extra stuff to put into a paper to at least try to get somebody, hey, maybe that's interesting. Yeah. Well, what do I do now? Look at that handsome devil over there. That was me, I don't know, 2 years ago, something like that? No, that was me when I was 18 years old, sitting in San Diego, going to boot camp for the Marine Corps. And I look nice and poised there. What I had just done was gotten out of a sand pit doing push-ups for 10 minutes while waiting in line to go take a picture. So I was covered in dirt from here down. What they do is they walk up. The dress blue uniform is split in the back. They walk up, they wrap it around you, they put the hat on, they say, look that way, and then they snap a picture. And that's how your Marine Corps pictures are taken. We all look like that, and if you smile, you're in trouble. Okay? But that's me. Right? So I did my combat tour in Iraq in 2003. I was actually one of the first people to cross the border from Kuwait into Iraq, went on the initial assault. We were in chemical suits the whole way up, all the way through, and I saw artillery going off and things blowing up and, you know, good times. What did it take? About a month to get to Baghdad, something like that. And I finally, after a month, got to take my chemical suit off. That was a smell. And my little brother actually did 4 combat tours in Iraq and Afghanistan, has a Purple Heart and things like that. He's okay. But yeah, so, you know, I feel the military thing is inside of me, and I care about it, and I actually care about PTSD a little bit. Because when I came back, I probably did some silly stuff that nowadays I would have been diagnosed with PTSD. Back then, it wasn't really on the radar. I was one of the first guys to go in and come back out. Some of the things that I did here that nobody knows about were probably PTSD related. Yeah. But about 4 to 17% of veterans coming back have PTSD, diagnosed. If I didn't get diagnosed, how many people do you think are not actually diagnosed? Yeah, it's a lot more. Well, right now, 22 per day commit suicide, veterans. Okay, a lot of that they think is linked to post-traumatic stress disorder. Yeah, it sucks. The rest of them end up with cardiovascular disease. You will die of heart failure or some other cardiovascular ailment 5 years sooner than somebody else who doesn't have PTSD. Okay? That's a big number. That's something I care about. So what do we know? We know that if we give a sympathetic antagonist, that's just blood pressure medication. It gets rid of the nightmares of PTSD. Because the nightmares are your conscious effort trying to feed back on the physiological problems that are going on while you sleep. You start getting scared, your blood pressure goes up, you get even more scared, but you're not awake to do anything about it. That's the nightmares. We control your blood pressure, no more PTSD, right? It goes away, okay? We know, or we started using that because we found out that in these Vietnam guys that have PTSD, they had high norepinephrine and epinephrine in their blood and urine in the mornings, right? So they're obviously hyperadrenergic, right? Their sympathetic nervous system's in overdrive. Well, that leads to, in somebody with PTSD, an increased baseline heart rate of 5 beats per minute. Just 5 beats per minute faster on average than everybody else. 5 beats per minute doesn't sound like much. Your heart can only beat so many times in your lifetime for the most part, and so, you know, that's taken away from the end. And their mean arterial pressure is actually 1 to 5 millimeters of mercury higher. Okay, they're starting to develop hypertension, okay? There are 2 FDA-certified treatments. They're actually coming out with more and more, right? The 2 FDA-certified treatments are both selective serotonin reuptake inhibitors, right? Those are depression drugs, okay? Depression drugs. So what does it do? It affects your ability to think. It affects your job performance. And it doesn't actually fix the cardiovascular things wrong with you. It just kind of makes you forget about them. I don't want to just forget about my cardiovascular problems or my PTSD. I want to fix it, right? Me, I'm very well adjusted. Maybe. Maybe. But so this, you know, we're trying to get some treatments out there. It's not the right answer. So I did a small study, or started to do a small study while I was up in Chicago. I tell you, having a kid can really throw a wrench into things, because in the middle of this study, we had a baby, so it slowed down. So I started to do a study, right? And I did it because there's that gap in the research. That thing I specialize in, that sympathetic nerve activity where I put into your leg, oh yeah, I started doing that in veterans. It's good stuff, right? And I also did it in the veterans coming back from Iraq and Afghanistan, the new veterans, right? Because all that previous research was done in Vietnam-era veterans. And it was done in the 80s and 90s, meaning that they had already had the PTSD for 20 years by the time it actually got measured. And if you think about it, guys coming out of the military are accustomed to exercising 3 hours per week, right? It's only about an hour a day, but it's mandatory. 1 hour a day, 3 times a week. Okay, that's exercise. Exercise is good for you. You should all know that, right? Exercise is good for you. Yeah. Well, most of them come out doing that 3 hours a week to nothing. A lot of them come out and they never run again. They don't go lift weights, right? And they sit around. And when you sit around, your body just starts to kind of, right? And maybe that elevates those cardiovascular problems, okay? So in my small little start of a study that I was doing, I took veterans and I tested them with some psychological tests for pre-diagnosis of PTSD, right? And I classified them. They all went to combat. They all went to Iraq and Afghanistan, things like that. And then I did those barrel reflex curves on them. That was that neck collar thing again, right? And so that neck collar, I can measure your heart rate and I can measure your blood pressure while I do these stimuli. And I can draw out these barrel reflex curves, right? So if you look, those 2 dots that are right in the middle mean, this is pretty much the mean arterial pressure. It's estimated carotid sinus pressure, but that's mean arterial pressure because it should be the same here as it is in the rest of your body, right? As long as you're laying flat. And so that's right in the middle. And if I do a neck pressure, it goes back this way and it goes up, right? If I do a neck suction, it goes down this way, right? Same thing for, this is estimated carotid sinus pressure and mean arterial pressure, right? So I'm measuring that. Anybody notice something about the red and the blue lines? Well, they are different. We already knew that somebody with PTSD, so the red line is PTSD. We already knew that they were going to have a higher heart rate, right? Baseline heart rate, higher. And we already knew that they were going to have a little bit higher blood pressure. Those lines actually make me really happy because the machinery that causes the reflex appears to be acting the same whether you have PTSD or not. Meaning that if I can get your blood pressure back down to where it's supposed to be, the neural function is still functional in both sets of my soldiers, Marines, airmen that I had, right? That gives me hope that all I have to do is find a way to, without interrupting the autonomic nervous system, bring blood pressure down. Anybody know a good way to do that? Exercise would be the first good way to do that, right? We can bring exercise, use it to bring those back down. But not everybody wants to do that. Everybody wants the pill. You know, you want to get a pill and things. My idea is the best way to do that is probably going to be diuretics. You just bring the blood volume down, you can bring the blood pressure back down, and then this person is not considered hyperadrenergic. And I haven't done that yet. I've moved back down to Victoria, so I'm going to have to start this stuff all over again. But that's going to be something I'm going to look into, right? So these are the people that along the way have helped me out. Right up at the IPL, that was my boss, Bo Fernhall. His wife, Tracy. She was the other, that was a weird, I didn't realize they were married until like 6 months later. Paul Fidel helped me along with all the nerve traffic stuff. Peter Raven was my PhD mentor. Mike Smith started me off on the nerve traffic stuff. I don't know what he did, but you know, he was there along the way somewhere. But those are the people that I'd like to thank for where I am now. Do I have any questions about anything I talked about? Nothing. Dr. Squires, I know you've got something. I've never heard you not ask.
First of all, I think you're onto something with the PTSD. And we have thousands of veterans coming back. And I'd like to see you get your lab up and go with that. Because if we do a non-pharmacological readjustment of the baroreceptors, oh my God. We have 70 veterans. I took you to the Veterans Center today. We have 70 veterans and or dependents on campus. About 35 are actual combat veterans, and about 50% of them supposedly had reported with PTSD.
Sure.
Here at Texas Lutheran. And we have such a hard time going to the VA in San Antonio and getting them their medicines, so they'll go without medicine for a while, then they'll kind of go bonkers, and then they get in trouble.
Yep.
So we have a special veterans court that puts into PTSD into the judge making a diagnosis. Now we've been able to get a PA, a physician's assistant, out here on a regular basis to get their medicines. The problem is, it's still the SSRIs and still those medicines that are, I'm not saying highly addictive, but highly dependent.
Yes.
So you're onto something with this, and I hope you continue that research.
Well, I'm going to try. There's some people that are really interested in this type of research. Lots of people that are really interested. You just have to convince them that you can do it. Yeah. Anybody wants to help me do it? I'm actually a director of the graduate program right down there at the University of Houston, Victoria. I've revamped it. It was broken before, but now that I'm the director, I have revamped it into a degree in biomedical sciences, right? And once you pass my core, and my core classes are not easy because I'm not just going to let anybody get through onto the second part where you get to help me in the human cardiovascular control lab. You have to pass my core first. But anybody who's interested in a master's degree and maybe not found a place to go yet, or wants to stay in South Texas, wants to go to a school that's maybe not very expensive, you know, comparatively. Some schools are really expensive. I'm trying to get graduate stipends and things like that. That hadn't happened yet, but that's part of my revamping. Shoot me an email, whitedw@uhv.edu. And we also have a guy who likes to go out to the beach and collect microbes and run metabolism studies on them. He's one of the other tracks, and a guy who's really good at bioinformatics and genomics. So those are the three tracks that we have right now. Eventually, I'm trying to bring the forensics track back in, but keep in touch. If you go on my website right now, don't trust it, okay? I haven't fixed the website yet because I'm still getting all of this through administration. And
He's only had 2 months to work.
I've only been there for 2 months, and I've already fixed it for the most part, so. But we will definitely be recruiting students to start in the fall. Cool. Anything else? Yay.
For more information, please visit tlu.edu.